Immune system is required for defending the host against infections. However, immune responses are themselves capable of causing tissue injury and disease. Injurious or pathologic immune reactions are called hypersensitivity reactions. Hypersensitivity reactions may occur in two situations. First, responses to foreign antigens, microbes or non-infectious environmental antigens may cause tissue injury, especially if the reactions are repetitious and poorly controlled. Second, the immune responses may be directed against self-antigens as a result of the failure of self-tolerance. Self-tolerance is the ability of the immune system to recognize self-produced antigens as a non-threat while appropriately mounting a response to foreign substances. Responses against self-antigens are termed autoimmunity, and disorders caused by such responses are called autoimmune diseases. Hypersensitivity reactions are classified on the basis of the principal immunologic mechanism that is responsible for tissue injury and disease. There are 4 different types of hypersensitivities. In this video, we're going to talk about type 1 hypersensitivity. Immediate hypersensitivity or type 1 hypersensitivity most often depends on the production of immunoglobulin E antibody against environmental antigens. Disorders in which immunoglobulin E mediated immediate hypersensitivity is prominent are also called allergy, and individuals with a propensity to develop these reactions are said to be atopic. Immediate hypersensitivity may affect various tissues and may be of varying severity in different individuals. Common types of allergies include hay fever, food allergies, bronchial asthma, and anaphylaxis. In individuals who are prone to allergies, exposure to some antigens results in the activation of T helper II cells and follicular helper T cells. Most individuals do not mount strong T helper II responses to environmental antigens. For unknown reasons, when some individuals encounter certain antigens, such as proteins in pollen, certain foods, insect venoms, or animal dander, or if they are treated with certain drugs such as penicillin, there is a strong T helper II response. Immediate hypersensitivity develops as a consequence of the activation of T helper II cells in response to protein antigens or chemicals that bind to proteins. Antigens that elicit immediate hypersensitivity reactions often are called allergens. 2 of the cytokines secreted by T helper II cells or by follicular helper T cells activated by the same antigen are interleukin-4 and interleukin-13. These cytokines stimulate B lymphocytes to switch to IgE-producing plasma cells. Therefore, atopic individuals produce large amounts of IgE antibody in response to antigens that do not elicit IgE responses in other people. IgE antibody produced in response to an allergen binds to high-affinity Fc-epsilon receptors that are expressed on most cells. Thus, in an atopic individual, mast cells are coated with IgE antibody specific for the antigen to which the individual is allergic. This process of coating mast cells with IgE is called sensitization, because coating with IgE specific for an antigen makes the mast cells sensitive to activation by subsequent encounter with that antigen. When mast cells sensitized by IgE are exposed to the allergen, the cells are activated to secrete their mediators. Mast cell activation results from binding of the allergen to 2 or more IgE antibodies on the cell. When this happens, the molecules that are carrying the IgE are cross-linked, triggering biochemical signals from the signal transducing chains of Fc-epsilon receptor. The signals lead to 3 types of responses in the mast cell: rapid release of granule contents, synthesis and secretion of lipid mediators, and synthesis and secretion of cytokines. The most important mediators produced by mast cells are vasoactive amines and proteases stored in and released from granules, newly generated and secreted products of arachidonic acid metabolism, and cytokines. These mediators have different actions. The major amine, histamine, causes the dilation of small blood vessels, increases vascular permeability, and stimulates the transient contraction of smooth muscles. Proteases may cause damage to local tissues. Arachidonic acid metabolites include prostaglandins, which cause vascular dilation, and leukotrienes, which stimulate prolonged smooth muscle contraction. Cytokines induce local inflammation. Thus, mast cell mediators are responsible for acute vascular and smooth muscle reactions and inflammation, the hallmarks of immediate hypersensitivity. Cytokines produced by mast cells stimulate the recruitment of leukocytes, which cause the late-phase reaction. The principal leukocytes involved in this reaction are eosinophils, neutrophils, and T helper II cells. Mast cell-derived tumor necrosis factor and interleukin-4 promote neutrophil and eosinophil-rich inflammation. Chemokines produced by mast cells and by epithelial cells in the tissues also contribute to leukocyte recruitment. Eosinophils and neutrophils liberate proteases, which cause tissue damage, and T helper cells may exacerbate the reaction by producing more cytokines. Eosinophils are prominent components of many allergic reactions and are an important cause of tissue injury in these reactions. These cells are activated by the cytokine interleukin-5, which is produced by T helper II cells and mast cells. Immediate hypersensitivity reactions have diverse clinical and pathologic features, all of which are attributable to mediators produced by mast cells in different amounts and in different tissues. Some mild manifestations, such as allergic rhinitis and sinusitis, which are common in hay fever, are reactions to inhaled allergens, such as a protein of ragweed pollen. Mast cells in the nasal mucosa produce histamine, and T helper II cells produce interleukin-13, and these two mediators cause increased production of mucus. In food allergies, ingested allergens trigger mast cell degranulation, and the released histamine causes increased peristalsis, resulting in vomiting and diarrhea. Bronchial asthma is most often a form of respiratory allergy, in which inhaled allergens stimulate bronchial mast cells to release mediators, including leukotrienes, which cause repeated bouts of bronchial constriction and airway obstruction. In chronic asthma, large numbers of eosinophils accumulate in the bronchial mucosa, excessive secretion of mucus occurs in the airways, and the bronchial smooth muscle becomes hypertrophied and hyperactive to various stimuli. The most severe form of immediate hypersensitivity is anaphylaxis, a systemic reaction characterized by edema in many tissues, including the larynx, accompanied by a fall in blood pressure and bronchoconstriction. Some of the most frequent inducers of anaphylaxis include bee stings, injected or ingested penicillin family antibiotics, and ingested nuts or shellfish. The reaction is caused by widespread mast cell degranulation in response to the systemic distribution of the antigen, and it is life-threatening because of the sudden fall in blood pressure and airway obstruction.
Do not forget to like the video and subscribe to our YouTube channel.